Hello everyone, welcome to a new session on the applications of bioinformatics. In this session, we will study about the various applications of bioinformatics in basic research of biomedicine, microbiology and agriculture. Bioinformatics is the use of information technology in biotechnology for the data storage, data warehousing and analyzing the DNA sequences. In bioinformatics, knowledge of many branches are required like biology, mathematics, computer science, laws of physics and chemistry and a sound knowledge of information technology to analyze data. Bioinformatics is an interdisciplinary field that develops and improves upon methods for storing, retrieving, organizing and analyzing biological data. A major activity in bioinformatics is to develop software tools to generate useful data. Bioinformatics has become an important part of many areas of biology. In experimental molecular biology, bioinformatic techniques such as image and signal processing allow extraction of useful results from large amounts of raw data. In the field of genetics and genomics, it aids in sequencing and annotating genomes and their observed mutations. It plays a role in the text mining of biological literature and the development of biological and gene ontologies to organize and query biological data. It also plays a role in the analysis of gene and protein expression and regulation. Bioinformatic tools aid in the comparison of genetic and genomic data and more generally in the understanding of evolutionary aspects of molecular biology. At a more integrative level, it helps analyze and catalog the biological pathways and networks that are an important part of systems biology. In structural biology, it aids in the simulation and modeling of DNA, RNA and protein structures as well as molecular interactions. Bioinformatics research can be classified under three major approaches. 1. Analysis based upon the available experimental wet lab data. 2. Use of mathematical modeling to derive new information. And 3. An integrated approach that integrates search techniques with mathematical modeling. Current research in bioinformatics can be classified into 1. Genomics, sequencing and comparative study of genomes to identify gene and genome functionality. 2. Proteomics, identification and characterization of protein related properties and reconstruction of metabolic and regulatory pathways. 3. Cell visualization and simulation to study and model cell behavior. And finally, 4. Applications to the development of drugs and anti-microbial agents. Role of bioinformatics and its implications in various biomedical and related fields. In this particular figure, you can see the overview of various applications of bioinformatics. The following are some of the important applications of bioinformatics. They are microbial genome applications. In this figure, you can see the role of bioinformatics in microbial genomics. The revolutionary growth in the computation speed and memory storage capability has fueled a new era in the analysis of biological data. Hundreds of microbial genomes and many eukaryotic genomes, including a cleaner draft of human genome, have been sequenced, raising the expectation of better control of microorganisms. The availability of genomic and proteomics data and improved bioinformatics and biochemical tools has raised the expectation of the humanity to be able to control the genetics by manipulating the existing microbes. In the last decade, bioinformatics has been used for the microbial biotechnology in many ways. Computationally analyzing the wet lab data, genome sequencing, identification of protein coding segments and genome comparison to identify the gene function, the development of genomic and proteomics databases and inference of phenotypes, or example, higher level functions from genotypes or gene level functions. The major contribution of the bioinformatics in genome sequencing has been in number 1, development of automated sequencing techniques that integrate the PCR or bacterial artificial chromosome based amplification, 2D gel electrophoresis and automated reading of nucleotides. 2. Joining the sequences of smaller fragments or contigs together to form a complete genome sequence, and 3. The prediction of promoters and protein coding regions of the genome. Automated identification of genes can be done using the following ways. Number 1. Hidden Markov model or HMM based techniques such as Glimmer and GeneMark. Glimmer, which stands for Gene Locator and Interpolated Markov Model ER, is used to find genes in prokaryotic DNA. It is effective at finding genes in bacteria, archaea, viruses, typically finding 98 to 99% of relatively long protein coding genes. Glimmer was the first system that used the interpolated Markov model to identify coding regions. GeneMark is a generic name for a family of ab initio gene prediction programs developed at the Georgia Institute of Technology in Atlanta. Developed in 1993, original GeneMark was used in 1995 as a primary gene prediction tool for the annotation of the first completely sequenced bacterial genome of Haemophilus influenzae, and in 1996 for the first archaeal genome of Methanococcus janashi. The algorithm introduced in homogeneous 3-periodic Markov chain models of protein coding DNA sequences that became standard in gene prediction as well as Bayesian approach to gene prediction in 2 DNA strands simultaneously. Number 2. Searching the known database of genes such as GenBank to identify genes and the use of algorithms based on decision trees that identify start codons and stop codons of the coding regions. HMM based techniques develop multiple probabilistic state machines, each capable of identifying an ORF. Each machine predicts the next nucleotide character using a state transition with maximum probability and matches the predicted nucleotide character with the current nucleotide character in the actual sequence. Statistical training using known sample sequences derives the probability of state transition. In the case of microbial genomes, the HMM based software such as Glimmer has provided 95 to 97% accuracy. Identifying gene function. After identifying the ORFs or open reading frames, the next step is to annotate the genes with proper structure and function. The function of the gene has been identified using popular sequence search and pairwise gene alignment techniques. The 4 most popular algorithms used for functional annotation of the genes are BLAST and its variations, dynamic programming technique, Smith-Waterman alignment and its variations, indexing based scheme FASTA and its variations, and blocks that uses multiple sequence alignment of conserved domains to identify motifs or characterizing patterns of proteins. Pairwise genome comparison. After the identification of gene functions, a natural step is to perform pairwise genome comparisons. Pairwise genome comparison of a genome against itself provides the details of paralogous genes or duplicated genes that have similar sequence with some variation in function. Pairwise genome comparison of a genome against itself provides the details of paralogous genes, that is, duplicated genes that have similar sequence with some variation in function. Pairwise genome comparisons of a genome against other genomes have been used to identify a wealth of information such as orthologous genes, that is, functionally equivalent genes diverged into 2 genomes due to speciation, different types of gene groups or adjacent genes that are constrained to occur in close proximity due to their involvement in some common higher level function, lateral gene transfer, that is gene transfer from a microorganism that is evolutionarily distinct, gene fusion or gene fission, gene group duplication, gene duplication and difference analysis to identify genes specific to a group of genomes such as pathogens and conserved genes. Let us now study about the applications of bioinformatics in the field of molecular medicine. Although scientists mapped the genomes of various organisms in the laboratories, the greatest challenge for them was the need to store or compile the huge biological data. For this purpose, computers had to be used in the research field as there was no other way for the storing of the data. Bioinformatics provided this opportunity to the researchers to store the data in the form of databases on computers. The whole work of DNA sequencing, its observation, analysis and interpretation all was the work of computers and it could not be done manually. The complete sequencing of the human genome has enabled to improve the biological research and clinical medicine. Scientists can find cures against diseases either hereditary such as cystic fibrosis and Huntington's disease or acquired as in the case of cancer or heart disease. Due to the complete knowledge of human genome, it is quite possible to view or analyze only those genes which are directly related with the disease. It is now easy to observe the molecular basis of each disease. This whole step will enable the pharmaceutical industry to design drugs which will only target those genes which are diseased. The drugs which are present in the market can target only 500 gene products or proteins, but the mapping of complete human genome has enabled to use the computational tools and more drug targets and medicines which could only interact with those proteins whose expression is diseased without any or little side effects. Pharmacogenomics, a branch of bioinformatics, has enabled to develop more effective clinical medicines. Though there are some advanced drugs in the market, but they have failed due to their adverse effects in patients because of the differences of DNA sequences in different patients. Today, as pharmacogenomics is making advances in the science world, doctors should use drugs on the trial and error basis because sometimes patients showing same symptoms of a particular disease show different results against the particular drug. It is because their DNA sequences are different. In the future, it will be possible for the scientist to prescribe drugs after observing the genetic profile of an individual. If the genetic mechanism of a disease is known, then it might be easy for the doctors to take diagnostic tests more accurately. There are chances that in the near future, it will be possible to use genes instead of medicines to cure particular disease. Gene therapy is one of the techniques which will provide this opportunity. In the future, it will be possible to cure or treat the disease by changing the expression of the diseased protein. Let us now study about the applications of bioinformatics in the field of drug discovery. Bioinformatics has made it possible to sequence the genome of various organisms, and there are almost hundreds of organisms whose genome has been mapped so far. The database of these organisms are increasing in size day by day because every day they get new information about any organism. As other fields of science have made use of bioinformatics, similarly, pharmaceutical industry has also adopted bioinformatics and genomics for the drug targets and drug discovery. The understanding of molecular biology has made it possible to design and develop the drugs. In the beginning, whole animals were used to test the synthetic organic molecules in the organ preparation. In the recent years, bioinformatics has made it easy for the researchers that they can now easily target the molecules in the in vitro environment. Today, the pharmaceutical industries are capable of developing such drugs which are able to target 500 gene products. A gene product is the biochemical material which is produced from the gene expression. It is either RNA or protein. Scientists have now mapped the whole human genome, which consists of 30,000 to 40,000 genes. That is why it has provided the scientists to discover more drugs by observing the gene expression. Mapping of the whole human genome has enabled the scientists to have more drug targets, but still it is a big challenge to get the target which show successful results. It is the science of bioinformatics which focuses the attention of the scientist on the target validation instead of target identification. There are a lot of factors which should be considered during the drug target identification, such as nucleotide and protein sequences, functional prediction, mapping information, data of gene and protein expression. Bioinformatics tools help in collecting the information about all these factors. These tools accumulate the information in the form of databases. These databases save time, money and efforts of the researchers and save the information in the form of groups and subgroups. The information in the database also gives the knowledge of molecules. There are two ways through which pharmaceutical industries develop drugs, either broad spectrum drugs or narrow spectrum drugs. Broad spectrum drugs can be used in the form of antibiotics to kill bacteria which have pathogenic characteristics, but the positive point is that these drugs do not harm the human health. Role of bioinformatics in the drug target has now advanced to a great extent such that now DNA and protein chips are used. The chips are very helpful in determining the expression of proteins or DNA in cells. The clustering algorithms present in the bioinformatics tools help the researchers to make comparison of the gene expression data and distinguish the diseased cells from the healthy ones. These algorithms also enable the researchers to observe the function of the gene or protein during the process of disease. The process of designing a new drug using bioinformatic tools has opened a new area of research. In order to design a new drug, one needs to follow the following path. Number 1 is to first identify the target disease. One needs to know all about the disease and existing or traditional remedies. It is also important to look at very similar afflictions and their known treatments. Target identification alone is not sufficient in order to achieve a successful treatment of a disease. A real drug needs to be developed. This drug must influence the target protein in such a way that it does not interfere with normal metabolism. One way to achieve this is to block activity of the protein with a small molecule. Bioinformatics methods has been developed to virtually screen the target for compounds that bind and inhibit the protein. The next step is to study interesting compounds. One needs to identify and study the lead compounds that have some activity against a disease. These may be only marginally useful and may have severe side effects. These compounds provide a starting point for refinement of the chemical structures. The third step is to detect the molecular basis for disease. If it is known that a drug must bind to a particular spot on a particular protein or nucleotide, then a drug can be tailor-made to bind at that site. This is often modeled computationally using any of several different techniques. Traditionally, the primary way of determining what compounds would be tested computationally was provided by the researcher's understanding of molecular interactions. A second method is the brute force testing of large number of compounds from a database of available structures. Rational drug design techniques. These techniques attempt to reproduce the researcher's understanding of how to choose likely compounds built into a software package that is capable of modeling a very large number of compounds in an automated way. Many different algorithms have been used for this type of testing, many of which were adapted from artificial intelligence applications. The complexity of biological systems makes it very difficult to determine the structures of large biomolecules. Ideally experimentally determined by either X-ray or NMR, structure is desired, but biomolecules are very difficult to crystallize. Refinement of compounds. Once you get a number of lead compounds, computational and laboratory techniques have been very successful in refining the molecular structures to give a greater drug activity and fewer side effects. This is done both in the laboratory and computationally by examining the molecular structures to determine which aspects are responsible for both the drug activity and the side effects. Quantitative structure activity relationship or QSAR. This computational technique should be used to detect the functional group in your compound in order to refine your drug. This can be done using QSAR that consists of computing every possible number that can describe a molecule, then doing an enormous curve fit to find out which aspects of the molecule correlate well with the drug activity or side effect severity. This information can then be used to suggest new chemical modifications for synthesis and testing. Solubility of molecule. One needs to check whether the target molecule is water soluble or readily soluble in fatty tissue, and which part of the body will be affected if it becomes concentrated in. The ability to get a drug to the correct part of the body is an important factor in its potency. Ideally, there is a continual exchange of information between the researchers doing QSAR studies, synthesis and testing. These techniques are frequently used and often very successful since they do not rely on knowing the biological basis of the disease, which can be very difficult to determine. Drug testing. Once a drug has been shown to be effective by an initial assay technique, much more testing must be done before it can be given to human patients. Animal testing is the primary type of testing at this stage. Eventually, the compounds which are deemed suitable at this stage are sent on to clinical trials. In the clinical trials, additional side effects may be found and human dosages are determined. Let us now study about the role of bioinformatics in agricultural biotechnology. The sequencing of the genomes of plants and animals will provide enormous benefits for the agricultural community. Bioinformatics tools can be used to search for the genes within those genomes that are useful for the agricultural community and to elucidate their functions. This specific genetic knowledge could then be used to produce stronger, more drought, disease and insect resistant crops and improve the quality of livestock, making them healthier, more disease resistant and more productive. Comparative genetics of the plant genomes has shown that the organization of genes has remained more conserved over evolutionary time than was previously believed. These findings suggest that information obtained from the model crop systems can be used to suggest improvements to other food crops. Arabidopsis thaliana or watercress, Oryza sativa or rice, Triticum aestivum or wheat and sea maize or maize are examples of complete available land plant genomes. Application of insect genomics helps in identification of resistance mechanisms and finding the novel target sites. Genes from Bacillus thuringiensis that can control a number of serious pests have been successfully transferred to cotton, maize and potato. This new ability of the plants to resist insect attack means that the amount of insecticides being used can be reduced. By using in silico genomics, technology researchers can identify defense or disease resistance gene enzyme with their promoter region and transcription factor, which help to enhance the immunity and defense mechanism. Gene diet disease interaction of nutritional genomics aims to study the susceptible genes and provide dietary interventions for individuals at risk of such disease. Scientists have recently succeeded in transferring genes into rice to increase levels of vitamin A, iron and other micronutrients. This work could have a profound impact in reducing the occurrence of blindness and anemia caused by deficiencies in vitamin A and iron respectively. Scientists have inserted a gene from yeast into tomato and the result is a plant whose fruit stays longer on the vine and has an extended shelf life. Bioinformatics plays an important role to detect the metal from metagenomic sequencing obtained from contaminated soil. Several strategies for the identification, characterization and functional analysis of plant genes involved in triggering, signaling and response to biotic and abiotic factors have been recently envisaged. In silico biology plays an important role to understand the plant pathogen interaction at gene and genome of plants and pathogens. Let us now study about the role of bioinformatics in comparative genomics. Comparative genomics is a field of biological research in which the genomic features of different organisms are compared. The genomic features may include the DNA, sequence, genes, gene order, regulatory sequences and other genomic structural landmarks. Computational tools for analyzing sequences and complete genomes are developed quickly due to the availability of large amount of genomic data. Internet-based genome browsers provide many useful tools for investigating genomic sequences due to integrating all the sequence-based biological information on genomic regions. UCSC browser. This site contains the reference sequence and working draft assemblies for a large collection of genomes. Comparative genomic methods have allowed researchers to gather information about genetic variation, differential gene expression and evolutionary dynamics in primates that were indiscernible using previous data and methods. Ensemble. The Ensemble project produces genome databases for vertebrates and other eukaryotic species and makes this information freely available online. MapView. The Map Viewer provides a wide variety of genome mapping and sequencing data. Vista. Vista is a comprehensive suite of programs and databases for comparative analysis of genomic sequences. It was built to visualize the results of comparative analysis based on DNA alignments. The presentation of comparative data generated by Vista can easily suit both small and large scale of data. By comparing genes of different organisms, it is possible to trace evolutionary pathways by which one organism could have evolved into another. Such studies can not only throw new light on evolution, but also provide evidence for the migration of species, thereby bringing new evidence to historical and anthropological studies. Let us now study about the role of bioinformatics in DNA forensics. DNA profile of an individual called DNA fingerprint can help in identifying criminals, establishing family relationships, protecting rare wildlife species and matching organ donors. Forensic bioinformatics promotes establishing of forensic databases to store DNA sketches of crime scenes of known offenders and apply DNA testing. Strength in terms of accurate results is boosted by statistical and technological advances such as DNA microarray sequencing. Thin film transistor or TFT biosensors and learning algorithms based on machine learning. Genetic tests have been widely used for forensic evidences and mass fatality identification. Genetic testing results are integrated with information collected by multidisciplinary teams composed of fingerprint specialists, medical examiners and forensic pathologists. In several countries, new rules could allow fingerprints and DNA samples to be taken from anyone they arrest, whether they are charged or not. This will be certainly facilitated by the introduction of DNA chip based microarrays, and thin film based transistors. Hence, it is clear from this module that bioinformatics is a highly emerging field with a wide range of applications. Here is a consolidated list of the various applications of bioinformatics. Microbial genome applications, alternative energy sources, crop improvement, forensic analysis, bioweapon creation, insect resistance, improve nutritional quality, development of drought resistant varieties, waste cleanup, veterinary science, climate change studies, molecular medicine, personalized medicine, preventive medicine, gene therapy, drug development, antibiotic resistance, evolutionary studies, and finally biotechnology. Let us now summarize what we have learnt in this session. Bioinformatics is an interdisciplinary area of the science composed of biology, mathematics, and computer science. Bioinformatics is the application of information technology to manage biological data that helps in decoding plant genomes. During the last 2 decades, enormous data has been generated in biological science. Firstly, with the onset of sequencing the genomes of model organisms, and secondly, rapid application of high throughput experimental techniques in laboratory research. Bioinformatics develops algorithms and suitable data analysis tools to infer the information and make discoveries. Bioinformatics research is a multidisciplinary and vast area. It ranges from drawing a mathematical or physical model of a biological system to implementing data analysis algorithms to develop databases and web tools to access them. Application of various bioinformatic tools in biological research enables storage, retrieval, analysis, annotation, and visualization of results and promotes better understanding of biological system in fullness. To sum up, the sky is the limit and bioinformaticians have ample scope for research in specialized fields within bioinformatics. For example, informatics, cheminformatics, proteomics, genomics, etc. Here are a few questions for you to work out. Number 1. Explain the role of bioinformatics in automated gene sequencing. Number 2. How does bioinformatic tools assist in genome comparison? Number 3. Briefly explain all the applications of bioinformatics. 4. Name and explain some of the bioinformatic programs used in gene identification. 5. Describe the drug discovery pipeline and the role of bioinformatics. Here I can suggest a few books for your future reading. Bioinformatics, Concepts, Skills and Applications, written by S.C. Rastogi, Namita Menderita, Parag Rastogi, published by CBS Publishers and Distributors, 1993, first edition. Understanding Bioinformatics, written by Marketa Svelebille, Jeremy O. Baum, published by Garland Science Publishers, 2008. Bioinformatics, Databases, Tools and Algorithms, written by Orpita Basu, Siminder Kaur, published by Oxford University Press, 2007. Introduction to Bioinformatics, written by Teresa K. Atwood and David J. Parry Smith, published by Pearson Education Limited, 1999. Thank you for watching this program. Let's meet in the next session with another topic. Until then, bye.